So, without any further ado, I think I'll give you a short intro, Gaurav. I think people have stopped trickling in a little bit. For several months now, well, actually, for many sessions previously, we've been looking at different models of neural organoids and neural systems in vitro in general, and how CNS regionalization can be recapitulated um, in these systems. And I think Gaurav's is one of the most recent iterations of exploring this. So we've been talking a lot in the lead up to this and are very happy that you'll be sharing some of your past and most recent work with us today. Thanks so much, Gaurav. You can take it away.
Thank you very much. Just two couple of things. I really don't mind to be disturbed in between, so
Great.
we can have a dialogue. You can whenever you want to have a question, just unmute yourself and interrupt, no problem. Second thing, this is my first time giving a talk to this elite audience in on Zoom. So if you see my hands are moving and the mouse is not moving, then please stop me and remind me that Gaurav, this is not a hall and you need to be moving your cursor. Uh, that's very important. Uh, I can talk a little bit about myself first. I am Gaurav. I'm currently a postdoc in Dr. Mercedes Paredes' lab in UCSF. She is a fantastic neuroscientist who focuses on neuroanatomy and found a very rare nestin DCX positive and MGE cell population, which is also and we call them nests. And then and she's teaching me all the different brain anatomy, and now we are atlasing basal ganglia development. I did my PhD in Agnete Kirkeby's group in the NNF Center for Stem Cell Medicine in Copenhagen. That's where this whole work was done, where we developed this microfluidic system that can mimic or create a full-on neural tube in one shot. And that's what we'll be talking and discussing how we can use these stem cell models to basically study the earliest events in the human neural tube. Let's start with that. So the very long title is mapping early patterning events in human neural development using an in vitro microfluidic stem cell model. If you have a suggestion of cutting short this title down, please tell me. I'm all ears. But this very long title covers mainly two things, and that is studying the early patterning events and a microfluidic stem cell model device. So let's start from there. The major aim of my PhD and perhaps my career is to understand early human neural subtype specification. And with that, what I mean is that life starts from a pluripotent stem cell, and this gives rise to regionalized progenitor cells. And you're seeing a cartoon of the neural plate development side by side just for the context. And these regionalized neural progenitor cells will eventually give rise to the regionalized postmitotic neurons. That is neuron of forebrain, neuron of midbrain, and neuron of hindbrain. What we want to do is we want to understand what are the transcription programs that are responsible for such this early differentiation, and what are the lineage relationships between this guy to this guy to this guy and so on. And that we want to study in human context. And that's the current challenge, that human brain functions and develops differently, but most of this early neuropatterning information comes from model organisms, not from human itself. Though we need to give a huge shout out to these atlasing efforts who has given us this very gigantic transcriptomic map that we can now use, but still the obstacle remains the same, that the prenatal tissues are very difficult to obtain at that very early time points, which is 3 weeks, 4 weeks post conception. And secondly, human fetuses cannot be used for perturbation studies or any other studies. So there is always a need of modeling, and this will keep going on. So, what we can do? We can model neurodevelopment using stem cells. And what scientists have done before, they have created this fantastic system called organoids. What organoids can do, they are these self-assembling structures that can mimic the regions and the development patterns of the different areas of the brain. However, what they cannot do is to have a coherent design from forebrain to hindbrain in one shot. And to complement these organoid systems, uh, what we decided and some other labs decided to create a system where you can have a tissue from forebrain to hindbrain in one shot. And what you can do is you can harness the power of morphogens. So here you are seeing a cartoon of neural tube and the morphogens. What is a morphogen? A morphogen is a molecule that has a power to pattern the tissue. In terms of the neural tube, there are two main morphogens. One is the Wnt signaling and one is the sonic hedgehog signaling, where the low Wnt signaling gives rise to the forebrain area and the high Wnt signaling gives rise to the hindbrain area. And similarly, the high sonic hedgehog signaling gives rise to the ventral side of the neural tube, and the low sonic hedgehog signaling gives rise to the dorsal side of the neural tube. And what we can do is we can create this constant gradient of these morphogens in vitro and recreate. And there are four of my favorite models that I would like to talk or give the shout out to is the microfluidic models from Damers et al., Rives et al., and the diffusion based model from Scuderi et al., who is right here in the audience, and the model from Jiaping Fu's lab. We are here to discuss the model that was invented in our lab, that is Rives et al., and we begin from here. So what we do, we have a microfluidic gradient device, which is this Christmas tree-like structure that has these two inlet points. One is the 100% where you put the differentiation media with the molecule, and 0% where you put the differentiation media without the molecule. And then it mixes gradually and creates this exponential model of the exponential gradient of the molecule you want to create the gradient of. And then this gradient is then exposed to the cell layer embedded on the matrigel and grows up into a tissue which resembles a neural tube-like structure, and we call it MiSTR, microfluidic stem cell regionalization. This is a small video of how the device works, and I would like to share it with you guys. Uh, this is these are the injection pumps where the media is getting injected with the molecule and the one without. And here, when you go in, this is the assembled device, and in this little window, the tissue is developing, and all the waste media is then further pumped out here. And this is how the MiSTR system works. One thing that I forgot to tell you is that we were able to achieve this gradient at 160 microliter per hour, which we have tested with this fluorescent dye. So, having explained the concept, I want to divide this story into two parts. Or I would like to call it two episodes, episode one and episode two, where episode one is focusing on the modeling of the neural tube, and episode 2 is focusing on the mapping of the neural tube. In the first part of the story, I will introduce you to the two models with MiSTR can generate, that is the dorsal side of the neural tube and the ventral side of the neural tube. In the second part, we will get it more complex by introducing a third model, and that is the dorsoventral patterning in the forebrain region. In the first part, we have sequenced these time points 0, 1, 2, and 14 to establish how the system works. And then we have then further complicated by sequencing multiple time points and creating an atlas. So basically, in a nutshell, first part, establishment and the validation of the model, and second part is increasing the regional complexity and long-term culturing experiments, and then further creating a map and linear trajectories that we talk. So let's get the background of the story. This all started back in 2012 when Ignatiy Kirkeby, my mentor in my PhD, uh, was culturing these human pluripotent stem cells by giving them different dosage of the GSK3 inhibitor, which is Wnt activator, and she was able to achieve the forebrain, midbrain, and hindbrain identities. And when you throw sonic hedgehog in the media, you can also ventralize these cells in 2D. Now let's talk about Wnt activation or GSK3 inhibition right here. So when you look at the Wnt signaling, when there is no Wnt signaling, the GSK3 is then binding with the beta catenin and destroys the complex, and thus you do not have any transcription going on and nothing happens. But when the Wnt signaling gets activated, the GSK3 complex is deactivated, and the beta catenin is now free to go inside the nucleus and start doing the transcription, and thus the Wnt signaling will initiate all the downstream transcription cascades, and that would lead to differentiation. So what our aim was to establish to establish a constant gradient model using GSK3 inhibitor. So basically, we can bypass the whole wind signaling and create a neural tube. So we did that, and our gradient here in this MiSTR device is the GSK3 or Wnt activator. So from now on, I'm going to call it Wnt activation from here. So what we did is we grow this tissue, and then we cut into a half part, and then look under the microscope, and you see it's as early as day two. You see the tissue segregating into two parts. The forebrain area is marked by OTX2. OTX2 is my favorite gene. It marks the forebrain, and it always gives me the right direction on where the neural tube is differentiating towards. And then the complexity gets increased when you look at day 14, then all the other structures arise, like midbrain is marked by PAX8 and the diencephalon is marked by the NKX 2.1. Uh, also, uh, what we did is we take the MiSTR tissue, divided into five equal parts: A, B, C, D, and E. And mind you, the E region here marks the hindbrain side, and the A region here is marking the forebrain side. And this will be coming again and again in the talk, so I'm already giving you the geographical guidance of the tissue here. And then we do a QRT-PCR here, and see the reproducibility of the tissue. So what it does is you have all the forebrain markers in the A region, all the midbrain markers in the all the hindbrain markers in the E region, and all the midbrain marker in the C region. It The point I want to make here in this slide is we can create this tissue, and we can do so exactly in the same way again and again. What we have done is we have created a marker where if you have a tenfold expression difference in OTX2 and GBX2, you will see you'll see an 82.1% reproducibility every time we try the differentiation experiment again and again. So it is kind of reducing the stochasticity in the system. Also, this is a question that we were getting all the time, and maybe we have not focused this much in our previous talks, but after discussion with Ron and Carlo, I would like to bring this up, that MiSTR tissue is also reproducible in multiple lines. It's just that we need to adjust the key dosage, and we are able to achieve the patterning. And one thing that Ron pointed out, and that we never noticed it, most of this cell line based differentiation is actually happening in the midbrain regions, not in the forebrain. As you can see, in the forebrain, in A and E region, you'll always see FOXG1 and HOXs, but in the midbrain region, the midbrain region sometimes shift in B region and sometimes in C region based on the different lines. So there is that little line-based variation here. I there is hold on. Uh, Yeah, now good. So basically, yes, MiSTR model can generate progressive caudalization across the various PSC lines. And also, one thing that we were able to do is we could control the patterning of the dorsal and ventral side of the MiSTR tissue. So when we grow the MiSTR tissue in just the neural patterning media, it by default makes the dorsal side of the neural tube. And when we make the ventral side of the neural tube, we just add the sonic hedgehog in the media, and we get all these ventral markers in the neural tube, and also it maintains the forebrain to hindbrain regionality. And then we can take this dorsal and the ventral MiSTR tissue in the single cell data, and we can pull them together to create a whole neural tube transcriptomic data set. So my game or my role in this story is to validate the MiSTR model using single cell RNA sequencing. And this is my workflow, where what I do is I take this MiSTR tissue and I divide it into five equal parts. And then I dissociate into single cell and single nuclei. And what we want to do is we want to give the correctly patterned MiSTR tissue to the single cell RNA sequencing experiments. So we take half the part to cryopreservation and remaining half into qRT-PCR for QC. When these cells pass the QC, we pool them with the site-seq hashtagging antibody and go for the 10x experiments. What we want to do is we want to look for the cellular heterogeneity. We want to enrich for the differentiation trajectories, and we also want to generate the gene regulatory networks. And this is all we've been doing for all these 5 years. Uh, uh, so, so this is the question we were always getting: is that is rostro caudal organization caused by canonical wind signaling or other GSK3 pathways? Because we were basically hacking the wind signaling there. So what we do is we analyze the tissue at 24 hours post differentiation. And if you look at single cell data, it very clearly distinguishes between this OTX2 and GBX2 domain. And again, my best friend OTX2 marks the forebrain area, and the GBX2 is marking the hindbrain area. And when you look at the principal component of these two areas, you can see that in this hindbrain or the GBX2 positive cells, 12 out of 15 top loading principal component genes are the direct downstream targets of beta catenin. So that concludes that there is a pretty good chance that we are replicating the beta catenin signaling here and nothing else. Also, that time we were getting also a very cool thing, which I'll be talking more in the future slide, is the rostrocaudal patterning events. And what we want to show is that the rostrocaudal patterning is established prior to neuralization. What you're seeing here is the day one Mr. tissue where these OTX2 and GBX2 cells are very nicely segregating, yet we do not see any neuralization markers, for example, PAX6 and SOX1. And if you do an RNA velocity analysis here, you see these vectors are trying to segregate, but by day two, these vectors have clearly formed a boundary and organized themselves into two areas, which is the rostral and the caudal side of the neural tube. And until then, we do not see any neural specification yet. And that is mind blowing in my opinion, because what we have already heard or what we've already read in the textbook is that the first event is neuroepithelium and not the patterning. So that's very cool to study in the future. And also, one question that at that time we were getting is this similar to the developing mammalian embryo? And fortunately, from John Marioni's lab, the mouse gastrulating atlas came out. And if you look at the 24 hours MiSTR of OTX2 versus GBX2, and if you look at the mouse embryo at E7.5, they follow exactly the same pattern, that the cells are organized into OTX2 and GBX2. And also, all of these, all the all the genes in the caudal area are the same Wnt downstream targets like SP5, RBP1, and follistatin, which is also present in the gastrulating mouse embryo. So yes, it is similar to the RC epiblast patterning in this mouse, and that's the best that we could show back in 2019 at that time. Also, another question that I was getting is, can we identify these different neural tube regions using single cell RNA sequencing? We have shown this in the qPCR experiments, but the main deal was to show it at the transcriptomic level. And if you look at these two MiSTR tissues, which I've shown, which I've addressed before, that we are making a dorsal and the ventral side of the neural tube, and the neural tube here, you see that we get the forebrain marked by FSF1 and 6 in the forebrain in the MiSTR tissues, midbrain marked by PAX8 and LMX1 and LMX1A in the dorsal versus the ventral side, and then hindbrain marked by HOXes. And very interestingly, you get a very small population of SOX10 positive neural crest. And here we get the whole FOXA2 designated floor plate in the neural tube. And this is super cool because with just one gradient and one flow, you can make the whole thing, and you are getting the full on range of all these different markers from forebrain to hindbrain and in both dorsal and the ventral axes. Uh, and then this is my favorite, and that is, can the MiSTR neural tube recapitulate the features of secondary organizing centers? And now allow me to introduce to the ischemic organizer, or what we call the midbrain hindbrain boundary. What you're looking at is a mouse embryo at E11.5. PAX6 is a gene that marks the whole embryo but leaves the midbrain area behind. And then FGF17 very crisply marks this particular area, which we call ischemic organizer, or the midbrain hindbrain boundary. And if you look at the MiSTR tissue, uh, you'll see that PAX6 is marking everything but leaving a little area behind, which is then covered by FGF17. And when we first saw that, we were jumping. We were like, wow, because I can still very confidently tell you that there are very few in vitro models that can actually create FGF17 like this, and we were also able to do so. And now we could go back and solve this time tested puzzle of how midbrain hindbrain boundary develops, and that we will be addressing in the episode 2 of the story. And not just this, but the accuracy is so good that if you look at the ventral side of the neural tube, which is the ventral MiSTR, you do not see FGF17 expression. Now look at the ventral side of the mouse. There is no FGF17. So we are bang on perfect there. And that's where I was able to validate that this is there's a system, it works well, and then I can conclude with the first part that we created and validated a MiSTR model, which can be used as a proxy to study neural tube regionalization along both ventral and the dorsal axis of the neural tube. Second thing, single cell RNA sequencing indicates that the roster caudal specification is established before neuralization. And the main conclusion is the MiSTR model can recapitulate the mid-brain hindbrain boundary, something which is very difficult to obtain in an organoid system. So this all part was published in Nature Biotechnology a couple of years. And these are the two main guys who were the absolute geniuses and part of the story. He's Pedro, who basically figured out all the differentiation protocols, and he's Mark, who is the bioengineer who designed all these devices that we used even today to make the MiSTR tissues. And now let's come to the second part, which is where now we'll be having a third model, that is the forebrain model of the dorsal ventral patterning, and we'll be increasing the more complexity and try to fit in this puzzle. Uh, so let's just start with the third model, which is the DV4 brain MiSTR. And to that, we partnered up with Barbara Trideline's lab and Fatima. Fatima, I don't know if she's here today in the audience, uh, but it was Fatima and Charlotte who worked to figure out the perfect media condition, which is creating a proper gradient of sonic hedgehog and the Wnt inhibition. And then if you look at the MiSTR at the DA MiSTR at day 5, 9, and 14, you see that there are all the forebrain markers. And very interestingly, PAX6 marks the whole neural tube, and then gradually it slows, it restricts itself to the dorsal side of the neural tube. I'm going to be talking a bit detail in the coming slides. And then also we were able to create a smaller data set about it, which captures the very early events of the forebrain development, and what are the other complex cell types that are coming throughout the course of the MiSTR forebrain development tissues. And one thing that I would like to now talk about is that, as I said, that we grow we grow the MiSTR tissue in the device, which is packed, and it's a microfluidically device control. But the tissue grows and it need to outgrow the chamber. So we need to figure out how we can grow as a free floating culture. So what we do here is this is a day 14 MiSTR tissue which is now out of the microfluidic chamber. We take this tissue out and we cut this into like this half, and it's a very fragile tissue. You need to lift it up, so we made some custom tools here, otherwise it breaks. And then to reinforce the structure of the tissue, we coat it in the matrigel layer, and then we grow it on an orbital shaker like a normal organoid. And then this is how it looks at day 62. Fatima called it Mr. That's the name given to it by Fatima. And you can appreciate the size of this thing. Like, this is a small petri dish, and it maintains it even after coming out of the microfluidic device. It stays this 2 centimeter long tissue, and you can do all sorts of multiple experiments on this. Also, the same question that we were getting, not that mouse is not enough, so you need to prove it to human that all these regions of the MiSTR model can correctly map to the developing human embryo. And at that time, Emily Brown's data came out of the week 5 human fetal brain, which I also showed in the first slide. And this is a spatial data set here. And what we did was we mapped the MiSTR tissues of the different models on this, and you can see that these dorsal all these different models of the MiSTR tissue are mapping correctly to these respective areas of the neural tube. Like dorsal MiSTR matches or maps onto the dorsal side of the neural tube. Ventral MiSTR maps to the ventral side of the neural tube, and the DV forebrain or the dorsal ventral forebrain region of the MiSTR maps with the forebrain side of the neural tube. I can talk about if in later on how we did all this analysis, and how what models we train to do such kind of projections. But the good thing here is that combination of just two gradients, Wnt and sonic, are good enough to create these to create a rostrocaudal or a dorsoventral tissues that accurately maps with the developing human neural tube. So what I did was I went into lab and I generated a lot of MiSTR tissues and I did a lot of data analysis as well. So we created this MiSTR atlas. I mean, I don't like to use the word atlas. We created a MiSTR map which spans from day zero, which is pluripotency, to day 62, which is the late mature stages, and we were able to track the ever con the developing complexity of these different cell types throughout the MiSTR development. This data is now available on UCSC Cell Browser and on NeMO Analytics. And if you want to, uh, I just want to add one thing, that this is an integrated data here, but if you also want to have these individual time points, you can go to UCSC browser and explore them. We have annotated them. We have spent a lot of time on annotating those time points. That would be very helpful. Or if you want to do your own analysis, you can either text me. I can also provide you with these independent time points and analysis. So
Gaurav,
now
could
let's,
I ask you something quick here?
yeah.
So this is a little bit backing up, but I wanted to make sure we keep it in mind as you get us deeper into the single cell sequencing. Something you touched on earlier, and I remember in our previous discussions, you mentioned it as quite key. Could you tell us the selection process, or do you use the same selection process here before sequencing single cells, or was that just in that earlier system when you actually pull out populations? Could you tell us about how that plays into these systems and the data sets you made?
Selection as in sending cells to the sequencing?
Yes, the site seek step, is that
Yeah.
involved here too?
Yeah. I'm sorry.
Okay.
As I told you, this is my first time giving a talk on
No,
Zoom.
that's
So,
okay. I should have stopped you earlier.
here it
That's
is. So
exactly
this is
this.
this
Yes.
is the process we followed for each and every time and each and every experiment, because we wanted to ensure that everything that we sent to 10x should be robustly tested. And so what you do is you divide this tissue into equal parts, get isolate them into single cell and single nuclei. You save half of it in the cryopreservation. And Sam, you'll be happy to know we're using a cryostore stem cell technology media, and it gives you great results for sequencing when you bring them back to life. And then we do a qPCR experiment. When these cells pass the QC, which is I test them with 36 different genes from different areas of the brain, based on all the heat maps that I've shown you before, we hashtag them with the site-seq antibody, so we can later on track that which cell came from what region of the neural tube here in the MiSTR, and then we send it to 10x. So every time we are looking at a cell, in most of the tissues, we know that where these cells came from. Uh, Carlo, you're muted.
Thanks. So you sequence them all together, but have the ability to deconvolute later.
Yes,
Okay, so
you
but
can
there's
use
no selection process then. It's just tagging to know where it came from. Okay,
yeah.
sorry, I was misunderstood. That's great. Thanks for going over that.
Yeah, no, it's kind of necessary, because later on, I mean, in one of the puzzles that we solved was because of the hashtag antibodies that we knew where the cells came from.
Great, great.
So yeah, let's come to the atlasing part. So yeah, so now we have this full on data sets. I mean, in my PhD, I could not explore all of these different aspects. So I basically focused on two or three small stories. But it's freely available, and it's and anyone can take it and try to build their own research questions from here. So I would just go back to the temporal dynamics of the early tissue fate specification, and it's really close to my heart as well here. And what I've showed you before at that time in the previous slide, that at day one, you see that OTX2 and GBX2, which is the rostrocaudal segregation, and we don't see any neural markers, for example, PAX6 and SOX1. But then if you look at when you add more data, and then if you look at day five, Mr. Tissue, you see the first neural markers start showing up. That is PAX6 and SOX1. And not just that, it's very interesting that the first neural markers are also regionally divided. The forebrain neuralization is marked by PAX6, and the hindbrain neuralization is marked by SOX1. And these are the earliest. So there's not just one marker of neuralization. There are these two that also. So even the neuralization. in a way is regional. They are two different programs, and that's what I'm going to be talking in the next slide. So we wanted to explore this concept further. So what we did was we took the cells from day 0 to day 9, which were OTX2 positive, that is the forebrain lineage, and the GBX2 positive, which is the hindbrain lineage. And then we just did a simple pseudo time analysis, and we see that there are two different transcription programs that are guiding this, and all these green genes, which is OTX2, HSX1, and SHIZA2, they are driving the forebrain neuralization, and follistatin, SP5, and GBX2, along with SOX1, are driving the hindbrain neuralization. So these are these two transcriptionally distinct programs that are needed for the forebrain neuralization and the hindbrain neuralization. Uh, we wanted to test this concept in the 2D differentiation system. So what we did is we took Cole's pluripotent line, and then we differentiated them into neural lineage. How you do it? You do a dual SMART inhibition, same thing in MiSTR, and then we look for the qPCR time points, and you can see that NaNOG is a pluripotency marker that goes down very sharply, but OCT4 lingers, and at that time OTX2 and GBX2 are still there, and then all these genes which you are seeing here at the UMAP, when we differentiated them into a forebrain and the hindbrain lineage, which is the green is the forebrain and the red is the hindbrain lineage, you see exactly the similar gene expression pattern. And not just this, what we also wanted to test was that how robust is this early neuralization. So what we did is we took these H9 human embryonic stem cells, and we differentiated them into forebrain and hindbrain lineage. How do you do that? By if you do a Wnt activation, they become hindbrain, and if you don't do Wnt activation, they just stay forebrain. And then at day 2, we flipped the media, and we decided to convert the forebrain differentiation into hindbrain differentiation, and one just continue differentiating towards forebrain. And here in the hindbrain side, we try to convert the hindbrain differentiation into forebrain and continue, so on. But to our surprise, what we saw is that these cells are not just early neuralized, their fates are locked. So even after you flip the media, once a forebrain that is expressing these this OTX2 should be written here. So all these green cells are still expressing OTX2, and the hindbrain cells, once they got hindbrain identity, even you try to switch them to forebrain media, they will still maintain as hindbrain. And this is super cool, because not they're just not acquiring the identity, but there is an epigenetic lock here, that allows them to stay what they are, and we now are further exploring this at a methylation and epigenomic level here in UCSF, that what happens, or what are the factors that are responsible for such kind of epigenetic locks. And this will make a huge difference when you're trying to develop the protocols for translational stem cell therapies. So, long story short, I want to summarize these two slides, that perhaps there is no universal neural stem cell. What we knew so far is that there is one stem cell, or what I said in my first slide, that life starts from a pluripotent stem cells. That becomes a neural plate stem cell, and that becomes a rostral and the caudal neural stem cell. What we want to say is, no, that could perhaps not be true. However, there are two epiblast stem cells, and that are still OCT4 positive, but regionalized by OTX2 and GBX2, which later gives rise to the rostral presumptive neuroectoderm marked by HSX1 and SHIZA2, and then later on giving rise to forebrain and midbrain. And here there are the late caudal epiblast stem cells that eventually gives rise to the hindbrain neuroectoderm marked by SOX1, HOXA2, and CRABP2. Again, this is also, I want you to imagine in a scale that uh, you don't have to. Previously we are giving we're giving kheer for like 9 days to get them neuralized, but now you see that at day 2, since they already acquired the identity and they don't change, you don't have to give the kheer or the Wnt activation for that long duration. And when you're developing a product, that will be cost saving on a lot of kheer. Additionally, when you look at the forebrain side of the neural tube, what we did is we did an unbiased analysis of the Mr. tissue at the day 5 forebrain, and you see that there is no segregation between the dorsal and the ventral cell identity. But if you look at the RC ventral and you look at the A, B, and D plus E region, you see a very clear segregation of the rostral and the caudal cells. So that means that at day 5, the rostral caudal identities are established, but the dorsoventralization is still not done. So there is a delayed ventralization. And we looked more into depth and we tried to track the PAX6 and NKX2.1. And you can see that at day 5 and day 9, there is still a lot of PAX6 and hardly any NKX2.1. And then later on, at day 14 and day 21, the PAX6 and the NKX2.1 domain are further substantiated. And also in this day 9 positive cells, the expression of NKX2.1 begins, but it is beginning in this off-target diencephalic population, which is also marked by FOXD1 and FOXA2. So basically, what I'm trying to say here is that when you grow cells in the Wnt inhibition media, the cells are not susceptible to the cells are not responsive to sonic anymore. Uh, and and then we try to further replicate this concept in the 2D culture system, where we differentiated pluripotent stem cells into ventral telencephalic and the ventral forebrain identity. So ventral telencephalic is obtained by Wnt inhibition, and ventral forebrain is just no KIR, no XAV, and we end up getting the similar results where you see the PAX6 expression is really high in all the cell types, but then eventually it goes down. I'm sorry, there should be a legend for this, but it's not there. So you'll have to believe my word. And the NKX 2.1 expression here you see very high is in the diencephalic population and not in the telencephalic population. So I mean, you don't really have to add sonic until the time you are actually giving the Wnt inhibition or XAV in the media. And maybe Park Lab has already found out back in 2017, because in their MGE protocol, they start adding sonic at day 10, and before that, they just keep on patterning them into the ventral telencephalic lineage. Now, I wanted to further explore this question, which is the morphogenetic gradients capable of generating secondary signaling centers in vitro. And if you look at the whole atlas, and I've tried to compress it into certain cell types that are my favorite, and you can see that the MiSTR tissue endogenously expresses FGF8 and FGF17, marking the midbrain hindbrain boundary, BMP and SLIT, which are marking the floor plate, and the ventral telencephalon marked by Wnt 5A, Wnt 7A, and Wnt 7B. And mind it, all of this is done by just two gradients, which is Wnt and sonic, and I don't add anything on top of it. So I'll be focusing on one secondary signaling center, that is the midbrain hindbrain boundary from here. But these two are open for interpretation, and anyone can download the data and do another PhD on this. I was happy with stopping right here. So let's go back to the midbrain hindbrain boundary, and the question, which has been there in 90s, which has been there in early 2000s, and even still is there: where does the MHB come from and what it becomes? So MHB, it is midbrain hindbrain boundary, which is a boundary between the midbrain and the hindbrain, and this is exactly where you find in the neural tube. And here in the MiSTR, we could replicate it, and this is where we could also find in the MiSTR. And we thought that, okay, now MiSTR is a good tool that we can use and address this puzzle. So I need to introduce you a little bit with how the MHB signaling cascade works, and this is a great review from Worston Bailey Quiff that you all should read, which tells you how the isthemic organizer is developing. It happens with the juxtaposition of OTX2 and GBX2 domain in the mouse that eventually gives rise to the expression of PAX2 and the two sides of the midbrain hindbrain boundary, the rostral MHB, which is marked by the Wnt1, and the caudal MHB, that is marked by the FGF2. And later on, it becomes a more complex structure marked by all these genes, for example, PAX5, PAX8, and FGF8. So we thought, or what we asked is, can we actually replicate this model in the MiSTR system? And if you look at the MiSTR system in the single cell time point from the MiSTR, at day 2, we see this PAX2 population and OTX2 and GBX2 juxtaposition. Uh, At day 5, you see the first markers of the presumptive caudal and the presumptive rostral MHB comes out, and then later on by day 14 and day 9, the MHB is completely formed and marked by the FGF17 and FGF8 presence. Uh, we have always used FGF8 as the marker of the midbrain hindbrain boundary, and I want to emphasize the importance of the FGF17. So again, you're looking at the mouse embryo at E11.5, and you're seeing that FGF8 is marking both ventral and the dorsal side, while FGF17 is the marker that very crisply only marks the ischemic organizer, that is the dorsal side of the neural tube, which has the MHB. Uh, Mr. can do it. So basically, FGF17 has the border expression. It comes after FGF8, and this is the true real marker of the MHB, not FGF8. I mean, that could be a statement, but okay, I would like to stick on it for now. Uh, uh, so based on this and based on all of this knowledge, the
Can
question
I
is,
interrupt you there for a minute?
yeah,
It's great listening to you, and I appreciate your um, clear statement of facts that you think are interesting. But actually, I disagree with what you're saying here,
okay.
and I think it it FGF8. I'm not going to be able to produce all the facts, but I mean, I would say stretching back to Alex Joyner, we know that
Mhm.
FGF8 plays a very important role in specifying these different domains. I think that's true.
Of course,
And
yes.
I
I'm
think
never denying it.
so then I think what I'm finding interesting then is that when you say FGF17 is dorsal, and then you say quite assertively that it's marking the midbrain hindbrain boundary,
Hmm.
isn't it marking a state of the midbrain hindbrain boundary? And that there's actually and it's a sort of timing effect. The boundary. Uh-huh. I think this is a very interesting topic, and the interaction between
Yeah.
FGF8 and FGF17 is a. I wish we had John Rubinstein on the call because he I think would say something about this here, about how these two different FGFs are working. Anyway,
Mhm.
I thought I should just show that I'm still a sentient being. Thank you.
Well, yeah, I
No,
think
no, I
it's
think,
also interesting that not only is the dorsal ventral pattern of the two FGFs different, right? The 17 is limited to the dorsal, but even the rostrocaudal extent, where FGF8 appears to be a pinpoint, both dorsal and ventrally here, at least in that those first panels, and the 17 actually has quite a longer range rostrocaudally, even though it is still all midbrain hindbrain border. The FGF8 domain is very small compared to the FGF17 domain.
So let me make another comment then. So the ventral domain of the midbrain, hindbrain, I'll say of the midbrain is
yeah.
really critical to the topic that you're just going to come on to, which is making dopamine neuron. And so one possibility is that the FGFA is doing something different from the FGF17 in this structure. So maybe the question to ask you is why do you say so assertively that it's FGF17
Hmm.
that's that's the midbrain hindbrain boundary?
Yeah, yeah, actually it's in the supplementary figure, and I think I should have also added in the slide. So this slide is only showing the dorsal side of the MiSTR, where you see that FGF17 is here in day 9, and then FGF17 consolidates itself very well at day 14. What we see in the ventral side, that FGF17 is also there at the day 9 of the ventral MiSTR. It just disappears at day 14. So to
To
make
make it just a little
dopaminergic
more
neuron,
complicated.
FGF17 is equally potent, and I'm coming to it in the coming slides.
Can
Well,
I
and
make a
also
comment?
jump in, Silky.
Yeah. So for FGF17, I mean, Ron mentioned about Rubenstein. I mean, 17 is also expressed in rostral pattern in center, both brain or ventral region. It's well known, and many data support that. And now, uh, uh, Nicola paper with the macaque transcriptomic also detect FGF17 in Rostra patterning center. I think in your sample here, you don't have really Rostra patterning center in the sample because it must be the tip of the forebrain region, right? And then ventralized, right? But that's actually rostral patterning center, that region is developed before the sonic capture induction. So in your system, you're not seeing it, I think. Yeah, that's my guess. But it's very interesting that, it is showing very, clear expression in mid-hindbrain region.
Yeah, I think I can form my statement then a little bit more diplomatically. So FGF7. So there are. I think what I was not able to communicate here with this is that there is a thing called early MHB and late MHB. So there is a thing where you can induce the MHB, and then there's a late MHB markers that maintains the MHB or further matures the MHB before it completely disappears. So what we were able to model is that we were able to we were able to get the induced MHB or the early MHB markers, for example, starting from PAX2 and FGF17, FGF8, and then later we get the proper late MHB, which is marked by FGF17 and FGF8. With FGF17 specifically. So isthemic organizer is a very dorsal phenomena. That's what I understand. And that is very specifically marked by FGF17. And that's what we also see here in the MiSTR, that it has a broader expression pattern than FGF8, and it is differentially regulated in the. So I think I should have added a day 9 ventral here as well, just to show that you see FGF17 at day 9 ventral side, but then it kind of restricts itself in the dorsal side itself.
I think you're sort of getting at the real solution here, which is that the midbrain hindbrain region contains both a ventral and and this thing you're calling the isthmic organizer is actually stretches up from the ventral to the dorsal domain. It seems to me, listening to you, that that's what you're saying. FGF17 is critical for the isthmic organizer.
Yes.
And that both FGF8 and FGF17 are playing a role in the specification of the ventral regions, the most ventral regions.
Yeah. What I'm saying is, in terms of modeling them in vitro, we generally it's easy to get FGF8 expression. It's very difficult to get FGF17 expression. And until and unless we don't get the FGF17 expression, we should not think that the modeling of MHB in vitro is complete.
Yeah, no, I think this is very useful actually. The zona limitans is something similar, and there are issues there that we don't fully understand, which also involves sonic hedgehog signaling and ventral domains and cells that stretch up from the ventral to the dorsal domain.
Yeah.
Okay, thank you.
Yeah, I mean, I'm generally good when I'm walking and talking, then I can like move my hands. It's really I'm finding a behavioral issue that it's very hard for a personality like me to sit on computer and do a talk. Ron, for that, I would like you to take you to a little bit of history lesson there, and I will also tell you a story by Irving and Mason in 1999, where they did this MHB experiments. And what they did was they took the rhombomer 1 cells and they topically transplanted in the midbrain region, and then they were able to get FGF8 expression. And also, if you fuse the rhombomer 1 and the midbrain, that also induces FGF8 expression. However, if you take the OTX2 positive diencephalon and the GBX2 positive rhombomer 2 parts and fuse them, there is no ectopic FGF8 or MHB expression here. So the whole theory of OTX2 and GBX2 juxtaposition should have worked here, but it just does not work. So the question is how to recreate MHB in vitro. And that's where my friend Matthias comes in the picture, and he did all these qPCR experiments where you could track the early MHB markers and the late MHB markers, early MHB markers being PAXs and the late being engrailed Wnt and FGF17. And we were always able to see the FGF17 expression in the MiSTR tissue, and we were trying to basically then deconstruct that what are the regions that is responsible for inducing MHB and then maintaining MHB. So basically inducing MHB, looking at FGF8 and PAXs, and maintaining MHB, looking at the FGF17. And that's where we combine these multiple combinations. And the question that we asked are what are the conditions required to create midbrain hindbrain boundary? So our baseline is the MiSTR tissue where we see the FGF17 expression. And we thought that maybe it's just the Wnt signaling. So you need the Wnt signaling that is corresponding to the midbrain area. So we tested in the 2D adherent culture Wnt signaling at different concentrations, and we were not able to get midbrain hindbrain boundary expression. So we thought that it could be just the 2D, and you need a 3D cell-cell interaction, and that would be good enough to induce and maintain the midbrain hindbrain boundary. So we created spheroid culture system. And with steroid culture system and the different concentrations of Kir, we were able to see that at 0.8, which is almost like midbrain, you need expression of FGF8, but FGF17 is still not there. So basically, at 3D, cell-cell interaction can induce midbrain hindbrain boundary formation, but not maintain it. So we thought, okay, now we need to make the model more complex and we need to fuse the OTX2 and GBX2 positive population. So we need to recreate the whole juxtaposition argument. And there we fuse the spheroids. So and we fuse them in multiple different combination, a midbrain fusion, a midbrain hindbrain fusion control. And then midbrain hindbrain fusion is the OTX2 and GBX2 positive population fusion. And then the midbrain hindbrain fusion supplemented by the Wnt expression or supplemented by the FGF8 expression. And we see that in none of the conditions we were able to induce the we were able to see the FGF17 expression. So basically, we can conclude that none of the single condition, that is the Wnt signaling or the OTX2 GBX2 juxtaposition or the 3D self-organization, is good enough to recreate and to create and maintain the midbrain hindbrain boundary in vitro. Basically, you need all of the three in a perfect manner. So you need the right Wnt signaling at the right level of the midbrain combination with OTX2 and GBX2 juxtaposition. And that MiSTR happens to have them all automatically. And you can create a midbrain hindbrain boundary. So there's not just one
Well,
component.
but this part I love it. It's super cool. But I think this mid-brain, hand-brain plus FGF8, I mean, there is some induction. You think that's not promising to you?
Could you
No?
please?
You think that's still right?
I was not able to understand the question. Could you please repeat it? It's okay.
I think the approach is super cool, and I agree your interpretation mostly. But when you said no other 2D culture and fusion experiment actually mimicking this MiSTR system, what I think is for that part, this mid-hind brain with the FGF8 is still a little bit promising. Yes, and then right. So what I'm saying is there is some trend. Don't you agree?
Yeah, I mean, I think okay, there is some trend, but it's not as okay. I'm
No,
getting.
but
Okay,
that part I
I'm
agree.
getting.
Yeah.
I was looking at the on the other screen and not here on the where you made the drawings. My bad. Um,
Mm.
okay, yeah. So this is the qPCR normalized data. This is not single cell RNA sequencing. Maybe I was not clear about it before. And second thing, when you do qPCR analysis, you will compare and we still think that this much of expression of FGF17 is not good enough as compared to what you are getting in the MiSTR. And I'm not saying that MiSTR is the only thing that can do it. That's not what I'm saying. What I'm saying is MiSTR happens to have all of these conditions that are favorable for the maturation of MHB. They're two very different things.
Can you just expand on that a bit? I mean, why does it work in MiSTR?
Yeah, so why does it work in MiSTR? Is because when you look at these fusion experiment, so a midbrain and hindbrain fusion is essentially fusing OTX2 and GBX2 positive population. Now, if it is supplemented by Wnt or FGF8, if you do it only Wnt alone, or if you supplement it with FGF8 alone, you still don't get enough FGF17. MiSTR apparently happens to have indigenous FGF8 expression. It is getting Wnt because we are giving it Kir, and OTX2 and GBX2 juxtaposition is happening because it's a constant tissue.
Do you think it's something to do with the topography of the cells in the MiSTR setup?
Yes.
That
They
actually the cells are actually an epithelium and the epithelium's
Mm.
not disturbed.
I mean, that's really where you're getting the OTX2 GBX2 juxtaposition
How
naturally in the system, right? Is
yeah.
because of that continuous sheet.
Yes.
so here FGF17 from MiSTR was the data was cell wall bulk RNA-seq was done with the region, no, in the how can I say? It's not like you analyze all the tissue from the MiSTR. You analyze FGF17 in the behind the brain region sample,
Mhm.
right? And then here, how did you analyze for the other part with the fusion experiment? Did you all analyze all of them?
They're all qPCR. So everything here is qPCR. If
I
Yeah,
that
think
but
she's
makes
what's
sense.
asking about
what
the
what?
But,
position. Yeah.
yeah,
So
right,
Key is
right. But I'm
suggesting
saying,
maybe the
Okay.
sorry,
FGF17 signal's diluted because there's other
right.
cell
That's
types
what I'm
present.
saying.
Yeah, yeah, exactly.
Is this
FGF17 expression is diluted because there are other cell types. No, that's not
When
are
true.
you
you are
make a
you
Yeah,
taking the
spheroid
whole MiSTR?
culture,
Are you taking the whole
yeah,
MiSTR to do the
Mm,
qPCR, or do you chop it up?
No, no. It's like, so basically I introduced the methodology in the beginning that I cut it into five equal parts, A, B, C, D, and E.
Which
So
position
B,
are we looking at here?
so we're looking at position C, and
Ah, so right in the middle.
yeah,
Okay,
yeah,
trying to
okay.
catch the
I
got
think,
it.
yeah, okay. I thought I spoke it, but
yeah, it's okay. You're doing well. Don't worry. We're
uh,
asking questions. That's
Yeah,
okay,
allowed.
yeah,
no
this is
problem.
So
great.
Yeah, you see in the slide
yeah,
here.
yeah, yeah.
So I thought I made it clear that my bad, if
That's
I was
okay.
not
It's
able to
okay.
make it
Yeah.
clear. So in this slide, what I said is that this is here we get the midbrain hindbrain boundary in the neural tube, and in the MiSTR tissue, this is the area which is region C. Sometimes it can also be a little bit of sometimes it can also be region D because the tissue kind of shrinks in the culture, and sometimes we need to be very careful how we cut the tissue into five parts. We just use a tool that just breaks it into five areas. But mostly we find it into the region C. Sometimes when we find into the D, then we kind of call it still midbrain hindbrain boundary, but majorly it's the region C. That's what I wanted to say. So yeah, the conclusion here is that you need to have a constant gradient system.
What do you start with there?
right.
Hmm?
Yeah, here.
So what?
Okay.
Mm,
So what's a spheroid culture? Where do the cells come from?
Yeah. Spheroid culture. It's
right.
basically you
This is a
create
C1.
you.
Mm.
Yeah. Oh, that's the okay.
No, no.
Yeah. So the spheroid culture
That
is basically the pluripotent stem cell clumps which grow as organoid, like the way you will make a normal organoid.
I
Uh,
see. I
we'll just
see.
have a free floating clump of pluripotent stem cells, triturate with pipette. You put them in a non-treated plate, or you can also put them in under a ultra low attachment plate and let them grow in these different multiple combinations of Esbinogen and Kier.
And the adherent culture is similarly just starting from pluripotency.
Yeah,
The
all of them
cells
are.
are just, yeah, yeah, I get it now.
Are the
Yeah, I
spheroids and the fused versions grown first and then placed in the MiSTR gradient, or are they grown from
No,
the
no.
beginning in the gradient?
the fusion experiment.
Well, both the spheroids and the fused spheroids. When do you move a spheroid into the MiSTR, or does it start in the MiSTR gradient to begin with?
No. So we don't move
They're not.
anything.
Okay.
We fuse the spheroid at day six, uh, which is this midbrain hindbrain spheroids, because we believe from all the previous data that by that time the the regionalization is already done and the neuralization is also done. So that's the good time to fuse them. Spheroid culture, we don't move them anywhere. We just grow them and then take out the data at day 14. Adherent culture also, we just grow them. And MiSTR, we just grow it like MiSTR. All of them, they start from a pluripotent stem cell. the diff.
Yeah. Thank
Actually,
you. In your paper,
Mm-hmm.
if I were a reviewer of your paper,
Yeah.
I would want to see tyrosine hydroxylase on this schema.
Might
Ah.
come later, right? Pao has had
It
a
is.
question for a while here. Do you want to
It's
jump in here,
coming.
Pao?
yeah,
It's
I mean, it
coming.
was all linked to this discussion. I think my point was, have you tried to fuse organoids with a kind of the gradients from C and D and see if the boundary appears then, rather than
Yeah.
just start with pluripotency?
Okay. So C and D would be 0.8 and 1.0.
Yeah, because if I understood correctly, you say that Mr. is good at finding this boundary because of the right concentration of this progressive concentration of the gradient. So if
Yeah.
you took the ones too closer to that concentration, would you be able to generate it
Yeah.
in the
Yeah.
exponent?
I'm hearing you. Yeah. Uh, Yeah, we've not done that, and that's a very good point because our aim was to get a clean OTX2 and clean uh, GBX2. However, when we call this midbrain and hindbrain uh, organoid fusion, I'm not able to recall it exactly what key concentration was it, but midbrain is always, I know by default that it's 0.8, and hindbrain is generally 1.2 and 1.4 when we fuse them. So it is very close to CND, that I can assure you. But exactly CND, it's very difficult to say because then you'll have to divide the whole 0 to 2 gradient into 5 parts. And what is CND? 0.8 and 1.0 or 0.8 and 1.2 that we have already done here. Uh, I think you should also look for the micropatenting colony paper from R.A. Warmflesh lab where they talk about the wind timing that is responsible for the induction of FGF8. Uh, Here it's okay, how do I frame it? Here, when we added the wind, we also thought that you need this OTX2 GBX2 juxtaposition supplemented by the wind. So we don't want to remove the wind and try to recreate as close as what we go to C and D. It's very difficult to say that if we can actually fuse C and D. I would just say that if we are able to fuse 0.8 and 1.2, we are as close as what you are saying.
Can I ask you a question about that Wnt activation,
Yeah,
particularly as you've got Wnt1 in your panel here,
yeah,
and Wnt1 is clearly expressed in the roof plate.
yeah. It's a
So
rostral MHB marker, wind one and
yeah,
grid one.
it's in the roof plate,
Yeah.
and it's in the roof plate all the way down the spinal cord, right? So it's rostral. It's a major rostral marker in the caudal part of the nervous system. And the question I was going to ask you, well, in the mouse anyway, the question I was going to ask you is when you use your Wnt activation protocol, do you think that's mimicking Wnt1 in particular? I mean, I just don't know about the differences in the pathways that these different Wnts
Yeah,
employ.
yeah, this is also something what we were discussing last time as well. We are not mimicking all these independent winds. We are just mimicking the effect of beta-catenin, which is induced by wind signaling overall.
Yeah. Because I think when all of this discussion about FGF17
Yeah,
it's sort of coming into focus, it's very cool to be able to have such a detailed discussion together.
yeah.
Thank you.
I think so. I think with the help of this review, what I was trying to explain is that Wnt1 predominantly is the marker of rostral MHB, and FGF8 is predominantly the marker of the caudal MHB. So you can divide the mid-behind boundary into two parts. And here, these circles I made when I was making the presentation, but I never spoke when I was speaking, is that we were also getting the Wnt1 and FGF17, which is the rostral MHB and the caudal MHB, if that answers Ron your question. So
It's
it's
not so
not
much a
it does
question,
not go
really.
until the spinal cord. That's what I'm saying basically. It's very restricted to the rostral side of the MHB for now, at at the neural
Uh-huh.
tube level.
At this stage,
Yeah, at this stage, which
right?
is somewhat like week 5 to 6 post conception.
Wnt1 was the first gene to be knocked out in a mouse,
Okay.
and the phenotype was in the roof plate of the spinal cord. It wasn't in the midbrain.
Wow, that's good to
So
know.
I'm just thinking that there's something very interesting about Wnt1. And when you point out that Wnt1 is rostral in the midbrain hindbrain boundary,
Mhm.
that's something I wasn't aware of, actually. That's
Oh,
very interesting.
okay, that's good.
I think
That's
another
great to
thing
know.
that I'd say about all of these markers, and it sort of came up right at the beginning of your talk when you talk about morphogens,
Yeah,
it's it's one has to be very careful to sort of realize that this is a highly dynamic system.
yeah,
And the morphogens are acting in one way at one time and perhaps in another way a few hours later. And so, I mean, I think this is a great story. So I'm not being critical at all. But I think that here, I think it's astonishing that you can achieve so much with the premises that you're using, actually. I would love to know if tyrosine hydroxylase is expressed. Tyrosine hydroxylase is expressed at very early times in the ventral midbrain, and the cells make dopamine. And
Carlo, can you
dopamine,
open the
you could
UCSC
argue, is a
browser?
morphogen. Sorry?
Oh, sure.
I mean, Carlo can open the UCSC browser and he can look at all these time points. It's already open, so I mean, you can already figure it out. I mean, we have always seen TH with the context of Parkinson's disease because we are a stem cell therapy lab and helping the PD patients. But at early time point, TH, no, we were more careful about looking for markers like brachyury, noto that they should not be in the diff, and they were not there. Uh,
Well, brachyury is very early, right?
yeah, and
So TH is not as early as brachyury. Sure.
no, okay.
But as soon as cells start differentiating in the ventral part of the midbrain, which is
Yeah,
very early, right?
yeah.
They're going to be TH positive in the mouse. Because you can stain for dopamine. Not only are they TH positive, they're making dopamine.
Yeah, I mean, I'm coming to a story about it actually in
Okay,
the next slide. Uh,
good. Okay.
I'm coming to that story.
Keep going, and I'll bring up TH while you keep going.
Okay. Yeah. Okay. Okay, so let's conclude this part of the story by making sure that the MiSTR model can recapitulate the progressive roster-caudal regionalization. Only two morphogen gradients, the wind signaling and the sonic hedgehog signaling gradient, are good enough to make the whole axis of the dorsoventral and the neural or the rostral caudal side of the neural tube. Very important. Perhaps there is not one universal neural stem cells. There are two, and they are epiblast. They are late rostral epiblast and they are late caudal epiblast. So the regionalization happens before neuralization, and we need to keep that in mind. The MiSTR model shows that the single gradient morphogens is sufficient to induce secondary organizing centers like midbrain hindbrain boundary. There are other secondary organizing centers which I did not explore a lot, but it can be explored by other people. But we were at translational stem cell therapy lab, so it was always important to see your research, the fundamental research in the with the classes of translational application. So how the MiSTR model has advanced our translational stem cell research. So this is where this paper comes. So what we have done in the product that is in trial with Novo, that FGF8 was used to create the caudal ventral midbrain, and the question that we asked was, can we substitute FGF17 to FGF8? So can we have an alternate protocol, or can we have a better protocol for the stem PD or for the stem cell therapy products? And that's where Amalia came in, and Amalia did all these experiments where she substituted the FGF8 and FGF17. And the answer is yes. FGF17 is as good as FGF8 to express all these to express all these caudal ventral midbrain markers, which is LMX1A and FOXA2. And this story is now published in Stem Cells, and you can go and read here. So when transplanted, these cells, Ron, express TH positive. They become dopaminergic neurons in the rat Parkinson models. Uh, Second thing that we were able to explore with the MiSTR tissue at the day 14 ventral tissue is that it expresses all these cell surface markers which are published. Uh, And then we found a novel cell surface marker APCDD1, which is sitting right in the area, which is what we call the caudal ventral midbrain in the day 14 ventral MiSTR. So what Ulrich did was he grew these cells on the different lineages, which is caudal ventral midbrain, rostral ventral midbrain, and the ventral hindbrain. And then he sorted the cells which are which had the surface marker called APCDD1, and then transplanted those progenitors into the rat model of the Parkinson's disease, and you can see the rotations coming down. So this was so important because all these regulating bodies, they want one surface marker for your product to be screened. And this is where APCDD1 was a marker that was then patented by Novo Nordisk for their stem cell Parkinson's therapy product. So when you explore these, so basically the point is when you explore these MiSTR data at early time at these different time points, you can actually find out really cool stuff that could be used to advance our translational stem cell therapies. Uh, and then basically the overall conclusion of this whole story is that there's a progressive caudalization, and then we can create the secondary organizers. Also, MiSTR is a good resource. Whatever we have put together is a good resource to download. And when you're looking for the when you're looking to develop differentiation protocols, please go on the websites, look for your favorite genes, and then use them in your favor when you are making a diff. And then we will be further with Mercedes Paredes working on the finding out the epigenetic factors that are responsible for that fate locking. And for that, we'll be studying the methylome of the early changing neural stem cells. And lastly, I'm very, very thankful to the development journal that they found our preprint very worthy. And Laura Pellegrini and their lab mates, they wrote a very nice op-ed on the article. And I wanted to shout them out for this generous honor that they gave to us. And then the most important slide of the whole presentation, and that is acknowledgement. So very big thank to Vignete, who gave me all this opportunity. Pedro, who invented the differentiation protocols, taught me how to make misters in the lab because I was mostly doing coding. All the KidKiwi lab, all the wonderful time that I've spent in Copenhagen. And now I have bought this. She's very generous to give this to me, and now I've bought this whole devices to UCSF in the Bay Area. So anyone wants to try the 2 centimeter long neural tube organoids, please talk to me, and you can come to UCSF and have a look at them. Our collaborators, Barbara and Fatima, who have been very great in giving all these comments and fine tuning the DV MiSTR protocol, and Fatima giving the term MiSTRoid after microfluidics. And then another big thanks to Mercedes Paredes, who find some talent in me and gave me an opportunity to work here as a postdoc, and also keep on while doing the anatomical research, keep on working on the early neural tube patterning, and then we have secured a Wild Neuro Hub grant to explore this. Perhaps there is not one universal neural stem cell theory in future as well. Yeah. Any more questions? Most welcome, and my apologies. Yeah, I'm more used to of speaking and standing and talking. It's very different experience sitting on a computer and giving a talk like that. Please go ahead.
Thanks, Gaurav. Rachel, you've had a question for a while. Jump on in.
Yeah, so I put this in the chat. Can everyone hear me, hopefully?
Yep, we got you.
Yeah, I guess one of my questions was in terms of trying to identify these boundaries organically, whether cutting the MiSTR differently would help you sort of establish the edges of those boundaries differently, or maybe I misunderstood, but I'm trying to understand. You were saying you cut it into A, B, C, D, E, but I was wondering if you just happened to cut it a different way, whether that would give you like slightly different resolution on where these sort of boundaries begin or end, or whether you just happen to capture it perfectly in C, for example. So I think that's my question, is where the sort of 5 cuts came from, and do you think you would learn different information with different cuts?
I knew. Thank you very much for asking me. So there is no such thing as perfect. Uh, and I wanted to show you all the video that I showed. It has all these different components that needs to be assembled. Okay, and this is this little window where we grow, and this is uh, 20 millimeter. What we have done is we have made a plastic device, not plastic. We've made a stencil, and we just use that stencil which has like 5 equal parts. So 20 divided by 5. That's the 2 millimeter gap. Sorry, 4 millimeter gap, and I'll just use a forceps and I'll just place it here, and it would cut. But the problem with MiSTR is it shrinks. So when I seed them, I'll just show you an image of MiSTR tissue here. Okay, look at this. Now, when I seed them, it was here to here, but now it shrinks a little bit, and it will. You make a different MiSTR every time; it will shrink in a different way. So sometimes what happens is that you can find this MHB in the D region, and then we look for the OTX2 and GBX2 positions in A and E to see that if there's a tenfold gap, and you see that okay, it has patterned properly, then we will consider that D region as mid-bin hand-bin boundary as well, if that answered your question. So it's a lot of experience. We have made this like hundreds of these tissues, and we have so much of data on qPCR that we can now look at it and say that if this patterned well or not.
Yeah, I think that makes sense. I think just by nature of doing it so many times and discovering it in different sort of, whether it might be in C or D, that does answer my question of it seems like you've been able to capture sort of where that it is, and it's not always in the same place, or it's in the same place relative to the tissue, but not your cuts have helped you find that. So that makes sense.
Yeah. I mean, I wish I was in lab. I would just sit with all these little tools and plastic 3D printed stuff that I could show you.
Yeah, no, that makes a
Uh,
lot of sense. Awesome. Thank you.
yeah. I mean, for me, it started as just an atlasing project as a bioinformatics student, but then I just fell in love with patterning, and I kept on digging more and more, and then there was never coming out of it. And now this perhaps there is not one universal neural stem cell theory. I think I was the other day I was thinking about writing to Soraya to even try uh,
Mm.
to validate that concept on the duo maps model as well.
Yeah, the two gradients are quite similar.
Yeah.
So, Key, you've had your hand up for a while. Jump on in
Yes,
here.
can I ask a little more question? So Ron mentioned this briefly, but do you see any tonal limiter intrathalamus
I
cells?
have not explored zonal emittance interthalamica. I don't know if I see it yet. Also, to be
Right.
very frank, you should also
Because
see
you
it.
have a telencephalin dicephalin with identity, right?
Yeah.
So it should be, it can be in the middle of there, right? So I think they are the, what is it, 63, 6 and the IRX genes are the
Yeah.
markers, right? So I think you can simply first check those genes and then see whether you have them. I mean, that's, I think, would be another interesting point, whether it can be induced by wind modulation, right?
Yeah.
And then I
So,
think for the rostra patterning center, I think since it looks like you don't have that in the system, but I think it might require wind inhibition. So you have wind activation, gradient, but if you
mhm.
somehow combine wind inhibition in the system, then I think very tip would be little bit more like a rostra patterning center identity.
Yeah. So
That
this, I think so that's the part we did with Fatima,
mm.
where the dorsoventral forebrain or the dorsoventral telencephalic mister was made by the gradient of sonic and wind inhibition. Is that
Oh,
good? Yeah.
right. But that's no, it's a separate experiment or from the beginning.
Mm-hmm.
F DVXs was tested separately by from the APXs experiment. So this one was also started from the beginning, from the pluripotency.
I I'm not sure I was able to understand the question. Carlo, could you help me with
Oh,
that?
sorry about.
No, no,
Uh,
no.
maybe just rephrase it again for us.
I
Okay.
think Soki is asking whether you applied the Wnt inhibition and the sonic hedgehog from the beginning.
Oh,
But
yeah.
as I understand it, that's the way you do all these experiments.
Yes.
Right.
Okay. In
Yeah.
this one, we do Wnt inhibition from day 0 to day 9, and
Yeah,
sonic we start from day 3 to day 14.
that's
What
right.
we have seen is
Yeah, you're right.
in this particular experiment, the biggest, my bad, the legend, I don't know how it got missed. When I was making the presentation, here. Okay. So here, look at this media composition. So when you want to make a ventral telencephalon, you do the Wnt inhibition from day 0 to day 9. Same thing in the MiSTR, and then sonic
Mm.
from day 3 to day 9. What we see here is this color map that NKX 2.1 in the ventral telencephalic diff is not going up at all. And if you look at the ventral telencephalic diff, is shooting high. So basically, what I'm concluding is that you don't really have to add sonic. I kind of like I did not talk about figure 5 of this paper because I thought we might shoot over time, and I can talk a little bit about it now, that the how the ventral diencephalon and the ventral telencephalon are very different when it comes to early patterning.
Yeah, so
And
here I think you can actually have rostral pattern center, I think.
Mhm.
Right,
Mhm.
because they are not really dependent on sonic hitchcock.
Right.
No, they're dependent
So,
on wing. So if you look at this off-target population right here, this is all
right.
diencephalic population, right? Where there's no Wnt inhibition, supposedly. They're all expressing NKX 2.1. So if there's a Wnt inhibition
Mm.
involvement, the cells are not really susceptible or they don't respond to sonic well.
Aren't these diencephalic
Okay.
cells?
Yes, they are diencephalic cells. They are off targets.
Right.
They are
Mm.
off targets.
Well, I don't
I
know why
have
you call them off target, actually. Why do you call them off target?
because we are growing this in Wnt inhibition environment, right?
Yeah, but still, I mean, that's just sort of the way you think about it, right? What the result is showing you is that in Wnt inhibition, you see
Mhm.
diencephalic phase.
Could be. Fatima can comment on it actually. Uh,
Don't
better.
you have all the single cell sequencing across these data sets?
Yes, I have it, and I also have a figure here which I did not talk in this chat. Is this particular, that we are able to we're able to recreate a very specific FOXA2 and KX2.1 population here in the rostrocaudal model of the tissue where we do not have Wnt inhibition.
And this is very, very interesting. And it's also very early.
Yeah.
Yeah.
Mm.
It's a very
Actually,
rare population. It was
Very
only
early.
reported by Maroof et al. In 2013 in a cell stem cell paper where they say that at the early time point in the ventral diencephalon of FOXA2 and NKX2.1 co-express, and then with time they basically divide into two separate domains, that is NKX2.1 positive diencephalic domain and FOXA2, the whole floor plate in the back. I did not talk about this
right.
particular figure because I thought like let's just focus on MHB today and don't.
It's a very interesting figure, actually. I wanted to just comment with respect to Soki's questions and
Mhm.
your fabulous data that one could take all the different organizers that are defined in the macaque by Nicola and
Yeah,
simply project them into the single cell sequencing data and look to see. Here, you dominantly use single genes to guide you through the system, but one could actually ask questions about
I think it's a great
or
idea. I think I'm going
organize.
to do it in the revisions actually.
Yeah,
Yeah, I think it's a great idea, and since the data is on NeMO Analytics, it's pretty easy to do now. I don't
I
have
think
to
that
go
would be
through the code.
super cool, actually, because
Yeah,
I think the discussion following that projection would be very interesting to me.
yeah,
Because I don't think it's going to be simple, particularly, but very informative.
yeah.
Could I make another comment? In this talk, you've emphasized it over and over again, this notion that there's two different kinds of neural pathway.
Yes.
And uh, yes, this thing, yes, precisely.
Yeah.
So actually, if you look in I think this is a very important topic, actually. But just to make a very specific comment about it, and then we could extend it if people are interested. If you look in the Pijuan Sala data,
Yeah.
you can see, I mean, literally by inspection, you can see the NMP population emerging, the neuromesodermal progenitor cell, right, which is on your right-hand side there.
Yeah, here it is.
And of course, at that time, there's a population of cells that are more anterior, that are still in the epiblast, that are delayed, to use another Briscoe term. So I think this notion that you're proposing here is actually all over the Briscoe data or the NMP data, and it would be very interesting to sort of talk it through together.
Yeah, I mean, I also did not show here because I had to like put both the papers in the talk. I compared it with the Pijuan Sala data here. So what I did was I took these early time points, which is E7.0, 7.5, 7.25, 7.75, and E8.0, and then took cells of the MiSTR tissue from day 0, which is pluripotent stem cells, to day 5, and then try to do a reference mapping using SCVI tools, which is an autoencoders based label transfer. And what you see here is that the epiblast cells at day 9, day 0, it maps very well. But even at day 1, there is 52% population that is still mapping with the epiblast cells, which is pluripotent. And then the amount of these rostral neuroectoderm and the caudal neuroectoderm are then increasing, and the epiblast position is
Nice.
and if you look at the gene expression pattern from Pijuan Sala at that early time points, which I've highlighted here, you see exactly the same. So, I mean, the thing is that it's not an evolutionarily wow thing. I think it's just we need to. With Briscoe has showed it in spinal cord long ago that spinal cord progenitors are already regionalized before neuralization. We needed to look into a CNS scope of the human, and there is no model that we can do at that early time point. Is that a good comment, Ron?
I think it is. I think this is a really fascinating issue because it raises the question about what is there such a thing as a pluripotent ground state, or is the pluripotent state combination of states in a where this you separate this thing out, the epiblast here, but
Yeah.
how do these things actually behave in the earlier condition? And just to reference a paper from Derek Vanderkoy that came out recently. I mean, what Derek argues in his paper is that when you reprogram cells, you actually reprogram them through a neural crest state. So that this NMP type neural crest posterior neural progenitor cell has and its position in the pluripotency landscape is actually still currently a very interesting topic. And I think that if you're interested in rostral fates, then actually the idea that there's actually in the epiblast cell, not just a neural crest type bias in certain cells, but a more rostral bias. The nature of those cells and the rostral bias becomes extremely interesting, particularly when you read papers that tell you that risk for psychiatric disease can be found in extra-embryonic trophoblast-like cells, which are even earlier.
That is gastric.
Right. So do you see what I'm trying to say here? I think this topic's really interesting. And yeah, maybe that's enough. Thank you.
Yeah, I think I will be able to answer this in a couple of years. We wrote a grant and I got a fellowship, so we'll be doing some single nuclear methylome sequencing of these early time points, and I'm going to be doing it on MiSTR and pluripotent stem cells, and I think it would be a good idea to also do the same on other models like duomaps and make a methylome map and see is this exactly what you asked, Ron? Is this a pluripotent stem cell? Pluripotent state is a multiple state, or is it something different? Uh,
Can I
it's
just ask you one more question on a slightly different topic? The thing that's so interesting to me about your whole presentation technically is the MiSTR procedure, and that you're
yeah,
growing a flat sheet of cells. And
yeah.
I said this before, this is an epithelium you're growing, and the cells are not changing their position relative to each other in the epithelium. And the
Mm.
epithelium has very special properties. It's the epithelium that assesses and implements positional information. It's not some abstract set of genes operating in hyperspace. They're operating in an epithelium, right?
Mm.
And what I wondered actually is a very sort of nerdy type question. Because when you set up this thing in the first place, does it sort of grow to have this dimension? I think you said 2 centimeters. Does it sort of grow progressively from a smaller thing to be 2 centimeters long? And when you cut an A, B, C, D, E section, how many cell diameters is that?
Yeah, so
Wow.
we plate it on the whole 2 centimeter. So when I'm seeding the
You
cells,
you plate it initially, yeah,
so the whole thing is it does not expands per se. It
right,
It
the
fills.
thickness increases because obviously the cells are dividing.
right.
But we deliberately try to keep it flat and basically ensure the laminar flow of the microfluidic devices to sort of have the gradient flowing properly, and it grows as big as like 60 to 100 microns thick.
So you're talking about 4 millimeters thick on average, right? If it's 2 centimeters chopped into 5 pieces, the
Yeah,
width is like 4 millimeters
yeah, that's what
ish,
I
ish. Yeah.
said. I answered to Rachel's question that it could be 4 millimeter equal parts. But what happens is that pluripotent cells, when they are seeded with ROCK inhibitor. They are like this. When you remove the ROCK inhibitor, they shrink. So what you're seeing here is a little bit of shrink,
Ah.
right? So our our device is not. It's just a uh,
The reason they shrink when you remove the ROCK inhibitor is that's when they're forming an epithelium. The ROCK inhibitor is keeping the cells in a sort of pre-epithelial to mesenchymal transition state.
yeah,
And it increases their survival. That's why it was introduced initially. But
yeah,
it's also, there's a lot of evidence showing you that it's also interacting with the EMT signaling pathways. So when you remove it, it's at that point that the cells start forming an epithelium, really.
yeah, yeah.
And of course, the epithelium is going to be polarized. And so the two different surfaces are going to have different functions. And actually, when you use your microfluidic device to pass fluid over this thing, I mean,
Mhm.
again, this is a sort of warm flash type question, but it, is there a sort of key? I mean, do you know that the fluid is actually just operating mostly at one surface?
Oh. I don't know. I don't know. I mean, I know that none of the cells are dying, so basically every surface is getting the required nutrients. That's the only thing I can say.
Uh-huh. A lot of cells die, do they?
No,
You
none
said
of them
none.
die.
Oh,
They
none of them die.
the only death you will see in the hindbrain region, and that is because of the Kier. Kier is notorious. It kills cells. Even if you do a 2D diff, you will see some death because of the Kier. Other than that, because of the media or because of the flow, there is no death. I think what I needed to emphasize is always I struggle when I present Pedro's part, which is the first three slides. The flow, the flow is important. If you go like for example 800 microliter, if you go like 1 milliliter per hour flow, it can erode the cells. So 160 microliter per hour is a very, very slow flow. It maintains the gradient, and it ensures that the cells are sitting there and everything is
But
the
doesn't
media
rip them
reaches.
apart
Yeah,
physically.
yeah, and the media reaches. Uh, I mean, assembling it takes like 3-4 hours, but when it's done, it's everything is like airtight. So if you look at the video, like this, we ensure that there's no bubble. Otherwise, bubble can destroy the whole gradient. Uh, and then like this is
Right.
where I just pause the video right here. Oops, oops. Can you Can you see these devices here? This is where the
Yeah.
embryonic stem cells are seeded before assembly. So we seed it like overnight with in ROCK inhibitor and these PDMS walls. And then this is the assembled device, which is bubble free and everything. And yeah,
So when you set this epithelium up in the first place, and your day zero,
yeah.
the cells are you don't ever see uh do you see notochord? Do you see other parts of the body appearing?
Now, big, I mean, okay. Also, one thing, when we are seeding these cells, they are still in pluripotent media. We have not started differentiating them. The differentiation only starts when the assembly is done.
I know, but I'm still going to ask the same question, I think, which is that, I mean,
Okay.
isn't it rather surprising that you take pluripotent cells and you differentiate them this way,
Mhm.
but all the cells are differentiating to neural fates?
Yeah, because you do dual smart,
Yeah,
which
dual
is
SMAD starts right when the Wnt starts. Right after seeding the pluripotent stem cells, the dual SMAD and the Wnt start together. Correct?
yeah,
Gaurav? Yeah.
yeah, yeah. So
Oh, so you need to put BMP in. You need to flow BMP over these things.
There you go.
yeah, that's why by default it becomes dorsal.
Thank you very much. Great.
Yeah. So
If you guys want to, I have the UCSC and NeMO up if you want to look at genes, but I also know we're now almost at 3 o'clock, so we could maybe make a plan to do that another time, or we could look at some of it now. What do you think?
Just show us one or two examples, Carlo, just so we get the idea, and then obviously people are volunteering to stay on the call.
So this is the UCSC site that Gaurav has up. Actually, I typed in a bunch of genes that we had been talking about. Ron, you mentioned tyrosine hydroxylase. This APCDD1 is the marker, sort of your endpoint for some of this, Gaurav. And Silke brought up the zonolimitans, and this is one of the genes from the Mikali paper in the monkey that it marks the zonolimitans organizer. So maybe we can start with tyrosine hydroxylase. So here in this map, it's a little bit hard to see, but I believe what we're looking at is there's some increased expression here. So the light orange is close to 0 and the black is the highest. And there's some black sprinkled in here. If we look at the same data set now on NeMO, it's a little bit easier to see, but a little bit faint. Here's where the TH is coming on. And as Gaurav, you mentioned to us, the differentiation trajectory is about like this, where neurons are showing up here. So, and we can also look at it by day down below. So this is the same map colored by day rather than by MiSTR model. And again, you can see the days go like this somewhat. So definitely there's a good bit of
Right.
TH expression in your system.
Right.
And if we separate it by day in this last view down here, this is the same data separated into the 3 different MiSTR models, and then within those days across here. So TH is a little bit low. We could have a better, say, visualization of these low levels, but it definitely looks like it's doing a little bit of this in these. And in here in the forebrain, the dorsoventral forebrain, it doesn't look like it's well represented as it shouldn't be, right? There's a little more in the more caudal regions there. So, and I also brought up this is the marker that you ended up on for which surprised me. So we saw the TH out here, but this APCDD1 is really expressed much earlier in a lot of these places. And let's see which models it's coming up in. Again, yes, not so much in the forebrain as you would expect, but much more in the rostrocaudal models that include that region.
And that's why I recommend to look at independent time points, because then you get more clear picture of these things. I mean, this big atlas is good for like OTX2, FGF, GBX2,
Right. No, I hear exactly what you're saying.
all the different neuronal markers, good enough. But if you want to look for like these very specific details, because ABCD1 is a surface marker for progenitor, TH marks dopaminergic neurons, and this is basically an protocol on steroids of basically what was a dopaminergic neuron protocol in 2D. So
Yeah.
Someone else also has a question. I just spoke in between. I'm sorry.
The NeMO link Gaurav posted on the chat is not working. So I'd appreciate it if you can share the link.
The NeMO link isn't? Okay, yeah, I'll just send this one right here.
Thank you.
Sure. And then, of course, you can replace the gene, but that should bring you to this same page. So that's TH and APC. Just for interest, this is the zonal limitans marker from the monkey study that has really quite robust expression. There's an interesting little nugget here that looks like it's in the ventral model. We'd have to go sort of looking to see where we can look, maybe at when down here. Maybe it's easier on this violin plot. Here we go. So it looks maybe most robust. It's actually early and looks like it goes away.
It marks everything. I have so much of this gene.
Surprising, huh?
It marks the whole neural tube in the early time points for day 1, day 2, day 5.
Crazy.
Just make a comment about that. So one of the issues that I think comes up when one's thinking about these problems is that because of the way the field is developed, you sort of think of organizers as being kind of separate things that act on other cells, and that these cellular states are always completely distinct. But actually, at early times, the organizer can be a very dominant state in the tissue. And so the fact that here, for example, we see TCF7L2 more widely expressed, it's actually sort of the question, right? It's not a problem, right? Do you see what I'm trying to say? All of these cells may have organizer type functions in them as a consequence of TCF7L2 function. It's not necessary, but it's highly expressed in these particular ventral progenitor cells marked here. Do you see what I'm trying to say?
Yeah. As the sheet tries to understand where it is, right?
Right, exactly.
Right.
It's the resolution of these maps that becomes the really interesting question.
Especially here in this system, we have a spatial information. So we know that where is a telencephalic identity, which part is little diencephaly. Once we get some idea, we are looking for in the between, right? In the cell. And TCF7L2 express cortical region and mostly diencephaly, a lot of population and zonal limiters. So you need to play with several markers. But I think it's a promising result, I think.
Yeah, exactly.
There is
I mean, I know, Paolo, from looking at this NeMO thing that you sent me yesterday evening, that if you put FOXA2 in your search, you'll get an interesting pattern, which might just help people understand how to look at these data sets. Help us all.
Come on. Oh, here it comes. Oh, look at that.
You see that? That's really beautiful.
Yeah, yeah, yeah.
Right. See, it looks almost exactly like red,
Which is
your
ventral.
ventral. Yeah, which is your ventral.
Floor plate.
Yeah, let's look at the
This little
timing
thing,
now.
this
Yeah.
little thing on the right-hand side here, I think that's what you call anterior floor plate. And actually, that's a question I had from reading your paper, which is, when you say anterior floor plate, do you mean diencephalon?
Yes, not Hox
Yes.
positive.
Yeah, yeah, yeah. I think that's an extremely interesting group of cells, actually.
Yeah.
They
So
give
here it
rise
is.
to all kinds of cells of great interest in physiology. And there you see it, right? Beautiful hindbrain, midbrain progenitors, but also in this thing you call anterior floor plate. And the timing is also lovely.
I spent a lot of time curating this data.
Well, it's
Sorry?
paid off. It paid off. It makes it understandable from our end as we explore now. This is super interesting here is that it looks like it disappears, right? It looks like there's 2 or 3 time points here in the middle.
Mm-hmm.
What is that? Looks like maybe.
That's an organizer. That's what organizers do.
They disappear.
They know when
Fabulous,
the
beautiful.
game's up.
I mean, I've
My
not
work
even
here is
explored
done.
the day 21, day 35, yet my story stops at day 14. There's so much of substance in this data. There can be, if someone just like, that's why I'm saying just please download it and make your own questions. Because my aim was to study the early patterning and my scope. But
Yeah.
they are
Yeah.
gone. All the organizers are gone by day 14. Day 21, the whole new thing starts, which is because now it's out of microfluidics. It does not have the laminar flow.
Yeah,
It
yeah.
starts
You
self-organizing.
should check out figure 1 in Nicola's science paper, where he defines all these macaque organizers.
Yeah, I've
Just
seen
figure
that.
1 is this wheel, and you'll see all the organizers are transient. But of course, that's a signature. Of course, you can look for individual genes, but his definition is done by dissection. Okay. Well, I hope people have enjoyed this. I have. Thanks so much, everyone.
Thank you so much.
Thanks, Rathore.
Yeah.
Maybe for in between now and the next session, I'll send a projection of those primate telencephalic organizer signatures projected into your MiSTR data on NeMO, because we have the macaque signatures there. So
That would be super cool. I think I can use
wonderful.
that.
Thanks so much, Gaurav.
Yeah, thank you.
And thanks everyone for staying so late and participating in the discussion.